Histamine is chemically for 2-amino-ethyl imidazole. The nitrogen atom at position 1 is called as tally, whereas the nitrogen atom at position 3 is called as pros. Now this is the structure. Now this histamine, it exists in two tautomeric form. We can have hydrogen at position number 1, we can have hydrogen at position number 3. Now in the molecule in which the nitrogen is present at nitrogen 1, it is called N-tally tautomer, and the other form in which the hydrogen is present in the third nitrogen is called the N-pros tautomer. So this histamine can exist in two tautomeric forms. One is N-tally, another is N-pros, depending upon the position of this hydrogen. If it is present in first position, it is N-tally. If it is present in the third position, it is called N-pros. Now, the chemical name, you can see that this is a 5-membered ring system. Okay, this is a 5-membered ring system. This is a 5-membered ring system. Now, 5-membered ring system with nitrogen at 1 and 3 position having N saturation is imidazole. So this is imidazole. Now, numbering system in this imidazole ring system, it can start from here 1, 2, 3, 4, or it can start from the other way around, but the correct will be starting from this 1, 2, 3, 4. So this is fourth position onto which this CH2 ethyl group is attached. Now on this ethyl group, at the second position of this ethyl group, we have an amino group. Hence the name 2-amino 4. This is position number 4. 2-amino ethyl imidazole. This is position number 4. 4. This is ethyl. This is amino. So 2-amino ethyl imidazole. Okay. Now this histamine can exist in ionic form. It can exist in uncharged tautomeric form. However, at the physiological pH, the N-tally tautomeric form is prominent. Okay, so this is the form which is prominent in the physiological pH. The active form of histamine at all its receptor is the monocationic form of N-tally tautomer. Okay, so the active form of histamine is the monocationic form of the N-tally tautomer. Now this is N-tally tautomer. N-tally means we have hydrogen at position number 1. This is histamine, N-tally tautomer. Now this is the monocationic form. We have a positively charged this, and this is the dicationic form. However, this monocationic form is the active form of histamine at all its receptors. Now this histamine molecule, it exhibits 3 pKa values of 5.8, 9.4 and 14. Coming to the metabolism of histamine, now this histamine can be metabolized by two pathways. It can be metabolized by monoamino oxidases or diamino oxidases, or else it can be metabolized by the action of enzyme methyl transferase. So, under the influence of these amino oxidases, this amino group is modified to form a carboxylic acid group, and the resultant molecule is imidazole. This is imidazole, and this is acetic acid. So this is under the influence of monoamino oxidases or diamino oxidases, histamine forms imidazole acetic acid. Under the influence of methyl transferase, a methyl group is transferred at the first position of this histamine to form N-methyl histamine. Now coming to the receptors of histamine. Histamine receptors are of 4 types: H1, H2, H3 and H4. Now all these 4 receptors of histamine have different distribution in various tissues. So for example, H1 receptor, it is present in the smooth muscle of bronchi, gastrointestinal tract, genitourinary system, cardiovascular system, adrenal medulla, endothelial cells and lymphocytes. H2 receptor is present in the myocardial cells and the cell membrane of acid secreting parietal cells. Now we will discuss in detail about these parietal cells and their role in the acid secretion when we will study about the H2 receptor antihistaminics, H2 antihistaminics. The third receptor subtype is the H3 receptor. Now this receptor is a presynaptic or autoreceptor in the CNS, and it is also present in the gastric mucosa. And then we have H4 receptor. Now this receptor is present in intestine.